Okay, so thank you for the invitation. Today I'm going to talk about the development of process-based comparative models for bridging micro and macroevolutionary speciation research, and I will explain later what I mean by that. But the whole purpose of this research program is to understand why we see the heterogeneous distribution of species richness that we see across the planet Earth or across different groups of organisms. So one iconic example of this heterogeneity is the example of the tuatara, which has only one representative species today. Its sister group, the lizards and snakes, have more than 10,000 species, so there's a huge heterogeneity in the number of species, even though these groups have the same age, and this type of imbalance in the tree of life, we see it all over the place in different groups of organisms. So if we want to understand present-day levels of species richness, what controls the number of species in the present is really the accumulation of speciation and extinction events unfolding through geological timescales. So if we think about two species groups which arose at the same period in time, what's going to detect how much species they have today is that the differences in their net diversification rate, so the balance between speciation event and extinction event. If we start at a given time and a group with low net diversification rates is going to accumulate species slowly and will end up having less species at present than a group with higher net diversification rates. Okay, so being able to estimate this rate of speciation and extinction is really key to understand patterns of species richness. And one way that we can do that is by adjusting stochastic birth-depth models to the phylogenetic trees of present-day species. So just that, that we're on the same page here, what we see are the trees, the evolutionary trees among present-day species here of all mammals, amphibians, birds, fish, and so on. And these trees represent the timing of divergence of ancestral species and the evolutionary relationship between the different species that we see today. And of course, these trees don't represent whole of evolutionary history because we're missing all the extinction events that we no longer see in reconstructed phylogeny. And so if we want to be able to estimate speciation extinction rates from this data, we need to adjust stochastic birth-depth models to this data. In this talk and in the research I'm going to show here, most of the time the trees are going to be assumed to be fixed. So we take that as data, we assume that the divergence times are known without any uncertainty, which is not true of course, but these trees also come from models of molecular evolution, but here we're going to assume that we know them without uncertainty, so we know which species arise from which species and their timing of divergence. So if we know that, what we can do is use birth-depth models, the same that are used in population dynamics, but here the birth events represent speciation events that happen with a given rate lambda, and the death events also arrive with a given rate mu, but here represent not the death of individuals, but the extinction of lineages. So here we have a birth-death process at the level of lineages, and we want to infer this full process, the succession of speciation extinction events from their reconstructed phylogeny. So here we sample species at present, we know the divergence time, we know the succession of branching events, but we want to reconstruct this full process from this data. And this can actually be done, and the idea there, the reason why we can do that is that the succession of branching events in a reconstructed phylogeny under a birth-death process is different. The expectations are different than the expectations in the absence of extinction. So this is illustrated here. Here we have in bold the reconstructed phylogeny, and in thin lines the full process. And we're going to follow the number of lineages in the reconstructed phylogeny, so the data that we can observe today through time. This is a plot that is called the lineage through time plot. And what we can show mathematically is that the slope of this curve here on a log linear scale is equal to the net diversification rate at the beginning of the history of the clade, and it tends towards the speciation rates towards the present. So there is information here in the lineage full-time plot on how much extinction there has been in the tree. So under this simple homogeneous constant rate birth depth model, we could see that from this simple plot of this lineage full-time curve, we could estimate the speciation rate from the slope here, the net diversification rate from the slope here. Therefore, we would have an estimation of the speciation and the extinction rates. And now, if we want to complexify the models and include some temporal or heterogeneity across time or across lineages, then we need to resort to more advanced statistical technique. And so we are actually able to compute the likelihoods of trees. So here I mean the likelihood of succession of branching events and topology under the birth depth process. So here is a representation of the likelihood surface for the birth depth process that shows again that this process is identifiable. So here we see that we have a single peak in the likelihood surface, and so we can estimate here the net diversification rate and the turnover rate, so the extinction rate divided by the speciation rate. So we can get estimates of these parameters from a reconstructed phylogeny. So since these processes were developed starting from the 90s, there has been a lot of further developments that allow to estimate how diversification rates vary through time. We have the example here of a model with time constant speciation rates through time, piecewise constant speciation rates through time, and also across lineages. So here we see models where different net diversification rates are allowed across the tree, and detections of particular places in the phylogenetic tree where there has been a burst of diversification. So today I'm going to be talking most about models with heterogeneous rates of diversification across the tree, since I'm interested in understanding why some groups of species are richer than others. Those are going to be the places where we have a shift up in diversification rates, and so we're interested here in fitting models with heterogeneous rate of speciation and extinction to phylogenetic trees. Okay, so most of the models that have been developed in the literature are models with an equivalent of a local clock, meaning that there's an homogeneous rate of speciation and extinction that is shared across closely related species, so across monophyletic groups, and there are places in the phylogenies where there are changes in diversification rates. Here, different diversification rates are represented by different colors. So these models have been properly developed, and we are now able to fit models with such local clocks and representing a shared speciation extinction event across a monophyletic group of species. The type in my group, we developed models that allow for finer scale variation in speciation and extinction rate. So this model we developed here is called the cladogenetic diversification rate shift model, and here it's how it behaves. What happens in this model is that we're going to assume that at each speciation event there is a variation in the speciation rate. So here we're not trying to find particular places in the phylogeny where there is an exceptional change in speciation and extinction rates. We are going to assume that changes happen at each branching event, and when speciation happens, we're going to draw new rates in a log normal distribution parameterized on the ancestral rates. Okay, so here's the definition of a model. We have the ancestral speciation rate here, and when we arrive at node I here, we draw the rates of the two daughter lineages in this log normal distribution, which is parameterized on the ancestral rate, lambda I, and there is a trend component parameterized by the parameter alpha, which allows for a temporal trend in the speciation or extinction rate. It can either increase or decrease. trees. And then there's a sigma parameter that controls the stochasticity of a process. So if we have a small sigma values, that will represent the case where we have little heterogeneity, little variation in speciation rates, whereas if we have large sigma value, we're going to generate a lot of heterogeneity. And what we're interested in, of course, is not to simulate this process, but to fit it to reconstructed phylogenies. And we were able to do that by computing the likelihood of a reconstructed phylogeny under this process and then integrating or estimating the parameters using Bayesian inference. So using the Markov-Chain Monte Carlo, we can estimate the hyperparameters of the model, which are quite few. There's only the initial speciation rate, the deterministic trend, the stochastic component, and a parameter that controls the relative extinction rate. So that was great to develop this model, and we were able to fit it to phylogenies of relatively small size, but one of the limitations is that it was quite slow to run. And so we started to think about other types of statistical inference techniques that can be used. And we started to develop a Bayesian data augmentation technique that now we use a lot to solve models that we were not able to solve before. So I'm going to explain quickly the principle of what this is about. So before, what we were doing was to find ways to compute the likelihood of the reconstructed phylogeny. Right? And to do that, we need to integrate over all the potential extinction events that happen during the generation of a phylogeny. And it's usually analytically quite hard to do this integration of all the unobserved events. And it's also computationally intensive. Usually you have a bunch of integrals that you need to integrate numerically and so on, and it's pretty slow. To the contrary, if you want to compute the likelihood of a full tree, so the tree where we would also see the extinction events, then it's super easy, right? We observe everything, and we can multiply all the likelihoods of all the events. So it's pretty easy and fast to do. And Bayesian data augmentation consists in augmenting the tree with simulations that are consistent with the tree that we see, with the empirical tree that we have, under the model, to compute the likelihood that we obtain and to update the parameters, to augment our tree with the new parameters and so on, and to integrate over the potential histories. So we don't integrate anymore only on the parameters, we integrate on the parameters and on all the potential histories that happened. So here's an example. If we augment here a terminal branch, so here it's a terminal branching event in the phylogeny, what we do is to augment this branch under our process. It's called the CLADIS here, the Clade-Genetic Diversification Redshift model. So it's exactly the process that I described before. So we augment our tree here, and now we compute the likelihood of this tree with all the observed events instead of computing the likelihood to see this one branch here, given that there could have happened a lot of different speciation events and extinction events that led to this one branch that we observe today. So that's easier to do and faster, and so we update these simulations, update the parameters, do that over and over again, and we obtain our posterior distributions of the parameters and also augmented histories of the tree where we have the extinction events and the estimates of the speciation and extinction rates directly on the tree. Okay, so if we do that, for example, here on phylogenies of birds, what we see empirically is that there is a wide variation in speciation rates across different lineages in the tree. So here it's hawks and hares. It's a pretty group of birds, but it's still, all birds. And you see that you can have more than 2 orders of magnitude variation in the rates of speciation across the branches of the tree. Here we have a distribution of these rates over the whole bird phylogenies, and there's groups of birds where speciation rates is more constrained. There's less heterogeneity in the rates, but some of them, there's a really, really wide variation. So we can, wonder like why there is so much variation and what drives these differences across species in speciation rates. What other groups did were to implement our CLADES model using SMC, so that they can also have, they are also able to compare the statistical support of different models when compared to data. And when they did that and compared the support for these for models with our model, so a lot of frequent shifts with small changes in diversification rates versus models where they have few shifts but large effects. So these models that I showed at the beginning that were existing previously in the literature, we see that on small trees, it's homogeneous models that tends to be supported because there's a lack of statistical information in the data to support more complex models. But as soon as we have bigger trees, then we detect heterogeneous rates of diversification. And when this occurs, it's always this CLADES model, so the model with frequent shift and small effects, that is supported against models with few large shift and large effects. So it seems that this model with gradual variation gives a better representation of what happens in reality when compared to empirical trees than other models. Okay, so just as a side, to say that this model with heterogeneous rates but small variation across the tree, it has now been implemented in what we call a full Bayesian inference. And so what this means is that here we no longer fix the phylogeny, right? I told you that I was going to fit models to fixed tree. Here the tree is no longer fixed, and we make a joint inference of both the tree and the diversification process directly from the sequence alignment. So it's a joint process, and in this case we can see what is the effect of assuming different ways to represent rate heterogeneity across the tree on the tree itself and on the datation of the tree. And here you see, for example, a place here in the phylogeny where this makes quite a lot of difference in terms of both inference of rates, more rate variation with our model, and also a difference in terms of phylogenetic reconstruction and phylogenetic dating. So it's pretty, even though most phylogenetic trees at the moment, they're reconstructed with homogeneous rate models, it can make a big difference on the reconstruction of the trees themselves and also on the inference of speciation and extinction rates. So we're pursuing on the development of such models with a small variation in speciation rates across a tree. So the CLADES model is a model where the rates change at speciation event, and now we developed a model against thanks to this data augmentation technique, which allowed us to treat this model, which was not possible to treat before by direct likelihood computation, is a model where the speciation and extinction rate can vary as a geometric Brownian process. So the log of the speciation rates is a Brownian process with a trend here, the trend alpha, driven by alpha. And we can also include extinction in this model, and we can either have extinction that is constrained or not by fossil information. So when we don't constrain the extinction rate with fossil information, there's different ways to deal with extinction. Either we can make an assumption that there's no extinction. We can treat extinction. We can assume that it's constant throughout the tree. We can assume that there's a constant turnover rate, so a constant ratio of extinction over speciation. Usually this is more realistic. Or we can also assume that the extinction rate follows the same geometric Brownian process. And with data augmentation, we can really deal with this model where both speciation and extinction follows geometric Brownian processes. but we can also constrain the extinction rate by fossil information. So these types of curve of how extinction rates vary through time can be obtained also using birth-death process, but fitted to paleontological data, and we can get estimates here for different mammalian groups of how the extinction rate varied through time. So if we do that and we use data augmentation, we start to see things that become more and more realistic in terms of reconstructing past history. So what you have here is the reconstructed phylogeny of all mammals. And what you have on the left is the data augmented tree, so you know that augmented with these unseen extinction lines, informed by fossils, and where we can really see groups where there has been a lot of speciation events, but a lot of extinction as well. And we know we only see a small fraction of species here, but there's a lot of these events of extinction that are reconstructed in the tree. Okay, so that's one way to incorporate molecular and fossil information by constraining the extinction curve to be the one that we infer from the fossil record. Another way that is even nicer is to do it, if I see if I cannot. Okay, let's see. No, this didn't work. Okay, well, let's see if I can go back. I just want to show like that illustrates, okay, one will be enough, that illustrates the data augmentation. So here now in our birth-death process, so far we have dealt with this birth-death process where we represent speciation and extinction events and how it proceeds through history, and we make the inference on the reconstructed tree. Now we had a sampling process along the way that represents fossilization, and so yeah, this process is called the birth-death fossilization process, and now we can make inference on speciation and extinction rates from this combined information with fossils directly included in the tree. And yeah, okay, I will put it again. But what we see here are, when we treat this model with data augmentation, different realizations of the stochastic birth-death process with fossilization. You see that it changes depending on where we are in the chain of the Bayesian inference, the reconstruction of the rates and of the trees that are given by different histories under this process. And so now we've implemented this fossilized birth-death process in both the case of this clad-ES model with rate variation that happen cladogenetically, and with the birth-death diffusion model where rate changes happen anagenetically along species, along lineages, along branches in the phylogeny. Okay, so but the main message of all that when we build these models is to see that when we feed them to data, be them phylogenies or be them phylogenies and fossils, what we find is, again, this wide heterogeneity in speciation rates across branches in the tree. And so we can, well, there's a lot of work that is developed to try to understand what are the abiotic factors, climatic variation, and geological context, and so on, or biotic factors, competition, mutualistic, antagonistic interactions, and so on, that can modulate rates of speciation and extinction, and also, of course, species-specific traits, right? So if we think about, in particular, traits like dispersal capacity are really likely to modify speciation rates, for example, right, with more opportunities for population structure when we have intermediate dispersal capacities and so on. So there's all these different factors, and we have models, so we have diversification models with rates that can vary as a function of past environmental variables. We have diversification models that can vary with different degrees of, where rates can vary with different degrees of competition or other types of interactions, or models that are called state-specific speciation extinction models that can test for the effect of species-specific traits. But this doesn't really go to the core of which processes really modulate the speciation and extinction rate. So if we think about abiotic and biotic factors and species-specific traits, yes, we can think about different ways that they can modify extinction rates, but eventually they would need to somehow influence demography to be able to influence extinction rates. So for extinction rates, it's pretty easy. We would think that, yeah, it's through the effect of demography that these different abiotic, biotic, and species-specific traits act to modulate extinction rate. For speciation rates, it's a little bit trickier. I don't think, does it? No, it doesn't work, right? Well, it doesn't matter. Okay, so if we think about how abiotic, biotic, and species-specific traits can influence speciation rate, then we really have to think about the speciation process, right? And the speciation process by itself, it's been, you know, the subject of a lot of work, and is a complex phenomena, but what people are focused on when looking at speciation is how does the evolution of reproductive isolation occurs, right? So for speciation to occur, you need to at some point have the start of a subdivision of population, and then you need to evolve reproductive isolation. You need to have a reproductively isolated population to generate new species. So there's been a lot of theoretical and empirical work focused on how can we make reproductive isolation evolve. However, when people in macroevolution have started to test the relationship between the rate at which reproductive isolation evolve and the rates of speciation that we can measure with the comparative approaches that I showed you, there has been this mixed support or lack of support for an association between speciation rates and these processes that we focus on when we think about speciation at the macro scale, so the evolution of geographic structure, genetic differentiation, or the evolution of reproductive isolation. So those are three examples here where there has been efforts to relate speciation rates at the macro scale to these metrics of population differentiation, genetic differentiation, and so on with little success. We conducted one such study trying to see whether we can find a correlation or coupling between species genetic diversity and speciation rates in mammals. So we would say why genetic diversity? Because we start, or why mammals? Because we start for these groups of species and for this measure, this intraspecific measure of genetic diversity, we start to have global scale data. So we start to have decent estimates of genetic diversity for a lot of mammalian species. And our initial hypothesis was that genetic diversity is what supplies the genetic variation on which selection can act to generate speciation. It can be positively correlated with reproductive isolation or with geographic structure. So we thought that we should see a positive correlation between genetic diversity and speciation rates. And yeah, so under these geographical or ecological models of speciation, we expect this positive relationship. And we found one previous study that tested for this relationship and found a very weak correlation, and if anything, more of a negative correlation between genetic diversity and speciation rates in fungi. Okay, so we collected this on GenBank, all this information on mitochondrial sequences to measure genetic diversity in mammals. We obtained the estimate of speciation rates using our cladogenetic diversification redshift model. So here is each branch is colored according to an estimate of a speciation rate of a lineage in the mammalian tree. And here we have estimates of two different estimates of genetic diversity for a bunch of mammalian species here. And what we found was similar to what they found with the fungi, but here with much more data to support this relationship. But we found a negative relationship between speciation rate and genetic diversity at the global scale in mammals. It's the blue line here, and across clades it's pretty consistent almost for all of them. We have this negative trend. Sometimes it's negative, sometimes it's not, sometimes it's significant, sometimes it's not, but we have this general tendency for lower speciation rates when genetic diversity is high. We thought that, so that's contrary to our initial expectation, and we thought that this could be related to an indirect link, life history traits or traits that would both affect speciation rates and genetic diversity in the opposite direction and would generate this negative relationship. So we tested, as we know that, a bunch of candidate traits that are supposed to potentially influence speciation rates and genetic diversity. So things like body size, range area, generation length, and so on. And we did find that those were correlated with genetic diversity a lot of the time, but not with speciation rates. And when we tested for the correlation between genetic diversity and speciation rate, accounting for these different traits, our negative relationship was still significant. So it doesn't seem like it's this indirect effect of these particular variables that generate this relationship. Okay, so am I late? No, it's okay. It's good. Sorry, I thought I had half an hour, and I was like, whoa. Okay, so yeah, that was not our expectation. So now, how can we explain this negative relationship? So one hypothesis is that it's not genetic diversity that limits speciation rates, right? Our hypothesis was that you need genetic diversity to have divergence happening, and that genetic diversity might exert a limiting effect on speciation rates, but it could actually be the opposite, where when we have a speciation, and if speciation happens frequently enough, then this could have an effect on the accumulation of genetic diversity, right? So it takes time for genetic diversity to accumulate in population, and if we have speciation events splitting populations and lowering genetic diversity, we could generate this negative relationship. Another hypothesis is that the relationship between geographic structure and reproductive isolation and genetic diversity is not a positive relationship, but is a negative one. So there are theoretical results that suggest that these species that have a high geographic structure would have a more frequent, Yeah, so that under some specific, I think it's under the continent island model, where we expect that actually genetic diversity would be lower under geographic structure when there's a lot of geographic structure or not. So it's a result by Whitlock here. So that's another hypothesis. And the third hypothesis is one where this could be explained by demography with species with small rather than large effective population sizes that would accumulate reproductive incompatibilities faster because of a reduced efficiency of purifying selection. So that's one possibility also, and in this paper here, they built a model where they showed that it's possible to generate this effect, and under this hypothesis, you would indeed expect that you would have lower negative correlation between effective population size, genetic diversity, and speciation rates. And so what this illustrates is that at the moment, what we have done to try to link macroevolutionary speciation rate with different measures, intraspecific measures of what can happen during the speciation process have been correlative. So we are correlating macroevolutionary speciation rate estimated from phylogenies with measures of the evolution of reproductive isolation, genetic diversity, population structure, and so on. And it's really hard to distinguish different hypotheses on then the patterns, the correlative patterns that we see. It's also difficult to understand what are the processes that act as a rate limiting step with these correlative processes, because we don't know, like when we measure something at present, which is what we do when we correlate present-day speciation rate with other intraspecific measures, is at which stage of a speciation process we are. And so if we measure, for example, genetic diversity in the initial stage of speciation, then we expect that if speciation is taking place, that there would be high genetic diversity, because here these species are not recognized as true species yet. And so there would be a high genetic diversity here within this species here. However, if we measure genetic diversity in newly formed species, so just after speciation, then we would expect to see low genetic diversity just after speciation. So when we use this correlation at present, it's really hard to know at which stage of a speciation process we measure intraspecific metrics of genetic differentiation, and so it's hard to draw conclusions. Another difficulty is the relationship between the speciation rates we measure using comparative methods and what happens during the speciation process. So in these birth-death models that I showed, speciation is considered to be an instantaneous event, right? So it just happened at a rate, and at this given rate, we have ancestral lineages that diversify into two descending lineages. It happens instantaneously and without any account for the different state of speciation. And when I showed this figure, what we can see that happens when we have speciation occurring is actually that we have first, we need to have an initiation of speciation, so we need to have something like a barrier forming or the colonization of a new niche, or polymorphism that occurs in sympatry to start the diversification process. Then we need to have this evolution of reproductive isolation, and then we need this to complete. So we need both the ancestral species and the incipient species to not go extinct until the speciation is complete. So there's really these three processes of initiation, evolution of reproductive isolation, and survival of both ancestral and incipient species until speciation completion to have a speciation event. So we're really, in this situation where here we measure an instantaneous speciation rate, and we have no idea which of these three major aspects of speciation drive the variation that we see in speciation rates, right? So we've tried to see whether it's related to differences in the speed at which reproductive isolation evolves, but there's much less that has been tried in terms of seeing how much differences there might be just in, speciation initiation that drives this heterogeneity, or differences in how much both ancestral and incipient lineages, at which speed they might go extinct. Okay, so one approach that have been developed to account better for these three stages of speciation in comparative methods is a model that is called the protracted speciation model, where here, instead of having just a speciation and an extinction rate as before, a simple birth-death model, there's a speciation initiation rate, but at this stage we don't yet have two good species. We have an ancestral species and an incipient species. Then there's the rate at which speciation completion occurs, so at which an incipient lineage can become good. There's a rate of extinction of a good species and the rate of extinction of an incipient species. So something that Pierre, who is a PhD student in my group, has started working on is to try to relate these equivalent speciation and extinction rates that we see in our original birth-death models with these different rates of speciation initiation, speciation completion, and extinction of ancestral incipient lineages. And here I put that in quote because here when we have this protracted speciation process, we no longer have an expectation of exponential distribution and so on, such as in the birth-death process. So it's really equivalent rates, and what we call equivalent rates here are the rates that are going to provide the same expected time for speciation and the same probability that extinction occurs. So when we do that, we can write this expression here in the two cases of the birth-death process and of a protracted birth-death process, and we find the rates of speciation and extinction that as a function of parameters of a protracted speciation model. And so then we can see how these different steps of a speciation process can influence the macroevolutionary speciation and extinction rates. And so those are the curves we have here with the 4 parameters of a protracted speciation model, and here the equivalent rate of speciation and the equivalent rate of extinction. I think the black curve is the analytical expression and the red curve simulation, so it's all good. And yeah, well, so what we see here is that the speciation rate, the equivalent speciation rate, is directly related to the rates of speciation initiation and the rate of completion, and it increases with the rates of extinction for incipient and good species, but only when they are high compared with the rates of speciation initiation and completion. And then the extinction rate, it doesn't depend on any of the parameters except on the extinction rate of a good species. Okay, so that's one way that we can start to see how the speciation and extinction rates that we measure at the micro scale from phylogenetic and potentially fossil information might be related to these rates, more specific rates that control initiation, completion, and extinction. Okay, so what would be great is if we could fit this model to phylogenetic data and get estimates of these rates, then we would have a better idea of what controls speciation and extinction rates and their variation across the tree. However, what has been shown before is that this, and we could have guessed, but this is not identifiable from only phylogenetic data. The speciation initiation and extinction rates cannot be confidently estimated, but we can only estimate the duration of speciation. But there's a possibility that if we add some information, so instead of having only the phylogenetic tree of present-day species, we have a phylogenetic tree of present-day species and some intraspecific information on population subdivision, then maybe these parameters can become identifiable, and maybe there's a chance that we can estimate these more specific parameters instead of the speciation and extinction rate from phylogenetic data. And then finally, this model that would be a better way to make this connection between macroevolutionary and macroevolutionary speciation rates, but it's still phenomenological. So here we're not modeling explicitly the interplay between demography, intraspecific genetic differentiation, and speciation, and so on. So we started doing that on 2 different ways, on 2 different projects. In the first one, we built on this cladogenetic diversification redshift model that I presented before, on which we add demographic processes. So here we have a population demography where species follow density-dependent population dynamics. We have growth rates varying along the tree as a Brownian motion process. And then at speciation event, we randomly split the populations into two subpopulations. And extinction naturally proceeds from the death of all individuals of a given species, or at least as soon as the population becomes small enough. And to this process, we can, if we do that, we have a record of a fluctuation in population size through time at the tip of a phylogeny. And so we can use some results from the coalescent process and population genetics to have expectations of what the genetic diversity of a species should be under this demographic process. And what we found by putting together this model is that we can produce both, with the model, we can produce both a positive or a negative relationship between speciation rate and genetic diversity. As a reminder, in mammals, we found this negative relationship, and we found that, so the slope of a relationship depends on the relative pace of speciation and accumulation of genetic diversity. So if we have rapid speciation, that's one of the expectations or the hypothesis that I gave before, where there's speciation is so fast that it can accumulate it can limit the accumulation of genetic diversity, and we have this negative relationship. So we can generate this process can generate this negative relationship, but now what we're doing is to fit this model to actual data, which we can do using some machine learning techniques. And then the second project that we have, trying to better connect these different crucial steps of speciation to actual macroevolutionary processes, so demographic processes and population genetics, is Pierre's PhD, and so he's presenting next. So you're going to see all about it just after. In conclusion, I'm sure this went a little bit fast, I'm sorry about that, but speciation and extinction rates vary widely across lineages, which is, ultimately explains why species groups, some species groups are much richer than others. We can link differences in speciation and extinction rates to species-specific traits as well as abiotic and biotic factors in the current models, comparative models that we have. So we have well-developed models to do that, but in terms of the models that allow us to really understand the demographic and genetic processes that act as a rate-limiting process in speciation, that remains something where there's a bunch of mathematical models that need to be developed. So with that, I want to thank all the people who've done the work, and thank you for your attention.